My name is Kiara Dooms, and this presentation is going to be over the role of glycoproteins and cell adhesion. A glycoprotein is an amphipathic transmembrane protein with hydrophilic regions exposed on the external side of the plasma membrane that have carbohydrate side chains known as oligosaccharides attached to them. They are formed by glycosylation in the ER and the Golgi apparatus. Glycoproteins are important for cell adhesion that forms long-term adhesions that forms tissues and organs. Here we have a picture of a glycoprotein. You can see the one on the left. They have the helixes that are part of the protein, and that's the part that's usually embedded in the membrane of the cell. And then on the side to the right of the protein, you can see the carbohydrate groups that are attached to it. And then the picture on the right is going to be a picture of a type of glycoprotein attached to a cell. And in this photo, you can see how the parts of the proteins come together to form the adhesion. I'm going to go over some of the location and functions of the major type of glycoproteins. First, you have the cadherins, which are plasma membrane glycoproteins that mediate calcium ion-dependent adhesion between cells. Next, you have the fibronectins, which are adhesive glycoproteins found in the extracellular matrix and loosely associated with the cell surface. They bind to the cells to the extracellular matrix and is important in determining cell shape and guide cell migration. Then you have the laminin, which are adhesive glycoproteins of the extracellular matrix. They are localized predominantly in the basal laminin of the epithelial cells. Lastly, you have the selectins, which are found in the plasma membranes, and they mediate cell-to-cell adhesion by binding to specific carbohydrate groups located on the surface of the target cells. A malfunction of glycoprotein adhesion is known to cause the following diseases. A malfunction in cadherins is known to cause cancer. When cells stop expressing the cadherin, it leads to cancer cells being able to easily metastasize and spread throughout the body. A mutation in the laminin causes junctional epidermolysis bullosa, or JEB, which is a painful, itchy, blistering disorder. Another blistering disorder is pemphigus, which causes painful blisterings just as JEB. A recent finding is that glycoprotein levels can be used as prognosis for cancer. A recent article entitled Study of VCAM1 Gene Expression in Normal and Tumor Tissues in Patients with Colorectal Cancer. So in this article, they looked at elevated levels of VCAM1 glycoprotein and said that they have prognostic significance in patients with colorectal carcinoma. So some background information is that the VCAM1 is normally not expressed. It mediates adhesion of the leukocytes and signal transduction. It's activated by inflammation. It is hypothesized to help tumor cells escape from the immune system when it is activated. In this study, they used samples from cancer patients. They took 29 males, 31 females, aged 28 to 81, and used reverse transcriptase PCR to analyze the levels of VCAM1 expression. They were able to show that the VCAM1 expression was correlated with the stages 0 to 4 of colorectal cancer. They also hypothesized that the VCAM1 could be a potential therapeutic target. A question about this topic is, what lab techniques could be used to detect specific glycoproteins in blood serum?